Thank you very much. I want to thank the Bangalore to this very interesting conference. And I am really impressed how many students have come here. And I have asked the people from the audiovisual support to make a microphone for you available so that we can really discuss the things that I'm presenting. So there are two different, challenging slots in a conference. Either you speak before lunch, everyone is hungry, or you speak after lunch. Everyone is tired from lunch. So I have chosen the second option and I welcome you here. I'm a neonatologist. This is a specialized pediatrician who looks after preterm babies and newborn babies. And you will understand why stem cell biology is very close to us. And you see here the parental advice of a stem cell that tells her daughter or son, you can be anything you want to be when you grow up. Well, you have already an understanding of stem cell biology that this is not true. So I will go with you through the cell type that is of most interest for us, the mesenchymal stromal or stem cells, the basic ideas that have been developed in animal data, the clinical practice we have acquired so far, and what the future will hold for us. So may I introduce you? This is the kind of patients I work daily with. This is Leonie. She is 25 weeks of age and has a birth weight of 500 grams. Or to put it this way, one handful of life, which has the potential to grow into an adult person. So you understand stem cell biology is at the core of this. Without stem cells, this would not be possible. What do you notice when you look at this picture? Can we have the microphone, please? So, who notices something on this picture? Who has witnessed a birth? Who is a parent? Okay, interesting. So I think India is in for a decreasing population. No one is witnessing a birth. You see, this is amniotic fluid, and this is pretty shady. This is infected. And that is one of the problems that preterm babies have. They are born out of an infection most of the times, and that kicks them out of their natural development. Here you see an MRI scan at 29 weeks of gestation, and here at 42 weeks of gestation. And all this development of the brain is now happening in a non-natural environment. An environment where you have high oxygen tension, where you have infections, noise, and a completely different day and night rhythm than in the uterus. So what influences this development of the brain? It is first of all infection, the inflammatory response, which may be triggered by infection, but also can be triggered by hypoxia. And do we have a window of opportunity to treat preterm babies? Yes. If a baby is growing poorly in utero because the mother is sick or the placenta is sick. But what if the hypoxic ischemic injury has happened, and then we have the so-called asphyxia, one of the most common problems after birth in all countries. And we have then brain injury resulting in cerebral palsy or disturbed development, learning difficulties, all things that usually don't present immediately after birth. I do not go into the lung as another organ affected by these developments, but about cerebral palsy. You remember from this morning's session that this is an umbrella term encompassing a group of non-progressive, non-contagious conditions that cause physical disability in human development. And please mind, again, development. And the incidence is pretty high, 2 to 2.5 per 1,000 live births. I do not know what the number of babies born in India is, but apparently since you haven't seen a birth and you don't have children, it doesn't affect you. So, what is the clinical manifestation? You can have a broad range, either from just one side of the body being affected, or like a cut-off, or you have all four limbs affected. And if you have that disease, you really need a good friend like a dog, because it really impairs your development and social interaction. And where do we want to go to? From here to here. So we want to improve the outcome. And the outcome is defined at a long time after the event. And that is why this is a team effort. If we do a good job at the neonatal intensive care unit for the first 4 weeks of life and don't ask how is the outcome after 6 years, we are missing the point. So here you see what part of the brains are affected that are then causing this delay in motor development. And you see how innocent this looks on an ultrasound scan or on an MRI, and how bad this is if you look at the loss of the cells and the calcification and the loss of tissue that you are then suffering. So what is the cause of the cerebral palsy? You have it during pregnancy because of prematurity, during the birthing process, and after birth. And look at the percentages. We are very much focused on this asphyxia event during birth, especially the lawyers who want to sue then the obstetrician. But this is only 5% of the underlying causes for cerebral palsy. Prematurity is much more important with respect to the numbers of patients and the incidence. Hasn't been there any progress? Yes. If you are born after 37 weeks of gestation, a so-called full-term baby, we can cool these babies for 72 hours and reduce their body temperature to 33.5 degrees Celsius. That is a profound change in the metabolism of those babies, but still 40% die or are severely disabled. So this is anything but the magic bullet. The preterm babies that present the major source of cerebral palsy, they cannot tolerate cooling. They are literally too immature and too fragile to tolerate this reduction of temperature. So there we are completely blank with respect to therapies. Well, I will go quickly through that because you have rehearsed this in the morning session. Stem cells are defined by their self-renewal and the rise to more differentiated cells. And the extent of differentiation is called potency. You have totipotent, pluripotent, multipotent, and you have rehearsed this in the morning. So which cell type might be of interest for these patients? We sat down several years ago and asked what we would like to have in a perfect world. We would like to fuel the endogenous repair mechanisms. We would like to stimulate proliferation of affected populations, anti-apoptotic properties, anti-inflammatory properties, and most important, the outcome as a function has to be improved. So what is known so far? Cooling and stem cells has been done, 23 babies, and they used autologous red blood cell reduced umbilical cord blood cells. So what do you have in umbilical cord blood? You have hematopoietic cells, you have some stem cells, you have a very interesting hodgepodge of cells, and they didn't differentiate this any further and gave it intravenously. And what is the outcome? They followed it up after 1 year, and more babies survived after the cells, and the survival was with a better neurodevelopmental outcome. You can imagine that everyone was very excited about that. However, what would be the perfect cell? The perfect cell would be the Doctors Without Borders, the so-called mesenchymal stem cells, that in other species can replenish a tail or even a whole limb. When they are called into action, they reiterate the developmental program during the development of the animal. We as humans have lost this property. However, the mesenchymal stem cells are the magic Doctors Without Borders that can do this in others. And we have learned that these mesenchymal stem cells have several functions in the brain. They rescue cells from apoptosis, they are anti-inflammatory, anti-proliferative, and they promote proliferation and maturation of the good cells that you need, and they inhibit those that are then the scar tissue or the inflammatory tissue of the brain. So what do we need now? We should know which cells to give at what time point to which patients in which dose and via which route. You can give it systemically. If you want to treat the brain, is then the systemic approach a good one? So now, since the microphone wasn't picked up, we will use your hands. Who thinks that giving stem cells intravenously is a good idea if you want to treat a brain dysfunction? Please raise your hand. Okay? Now one. No one. So who wants to give the brains directly into the brain? Ah, you. Good. Okay. Who wants to give the cells directly into the brain? Okay, one. Two. And what do the others want to do? Okay, we have to work on the silent majority. Okay, so these 5 questions need to be answered. And how do you do this? Well, then you have to do translational research, which means you take a problem from the bedside to the bench, and then from the bench to the bedside. Here is my bedside. You see my preterm baby. Here you see the experiment. And what is this? This is the tool that I use to mimic the clinical situation in a reliable and reproducible way. And this gives you an idea where you are in this. Laboratory research, translational research, clinical research, population research, public health. So we are really at the basics before we really do a big change and improvement for the public. So then now I show you where I work, because for one reason, not for the beauty of the building. Can you have a guess what those white dots on the green meadow are? Sheep, exactly. We keep them in a natural environment, and that makes them very happy. And I show you now the developmental biology, why we have chosen this model. Here you have the development of the lung, the gut, and the brain. You have sheep, human, and rodents. And if you look at the lung development, sheep and humans are very similar, and rodents are completely different. Gut development, same true. Cerebral white matter myelinization, same true. Sheep and human are pretty close to each other with respect to the developmental biology. And that is where I want to study my intervention. Here you see the differentiation of oligodendrocytes from pre to immature to mature oligodendrocytes in the human gestation and here in the sheep gestation. And this is the point where we have most of the problems with respect to cerebral palsy. So we are then building our model around this biologically defined moment. And this is, in short, the animal model. You take a fetus, you put ECG, EEG measurements on it, arterial catheters, and you put an occluder around in order to mimic the situation during birth when all of a sudden the blood supply stops because of a rupture of the placenta or because of a contusion of the umbilical cord. And here you see a 2x2 matrix. We have here the mesenchymal stem cells being given or not given, and here hypoxia, ischemia without treatment or with the stem cells. So you have 2 interventions, hypoxia, ischemia, and the stem cells, so you get a 2x2 matrix and 4 experimental groups. So, and this is how this looks like. This is fetal surgery, very delicate. You have to suture in the catheters. Here is the umbilical cord occluder, the most dangerous thing of the whole experiment. And we had to develop our own EEG and ECG electrodes because nothing worked in this fetal tissue. And then everything is put into place, and the mother is put into an individual cage, so she cannot start to chew on those cables and infusions that are coming out. And you see they are pretty happy if they are not alone. And what do we do then? We do an umbilical cord occlusion, and you see the blood pressure rises transiently and then falls down, and then you have a reperfusion injury. The heart rate goes down dramatically, so the perfusion overall is being impaired. And this has been described for decades. And here I show you the effects on the organs outside the brain. The spleen is reduced and becomes atrophic, the thymus in the very same way. This is what you would not expect if you just take out the blood supply and are interested in the outcome of the brain. And we looked at 2 regions, the subcortical white matter here and the hippocampus. So these are the 2 areas that we were interested in. And here you see again the 2x2 matrix. Here is shame with saline, shame-operated animals with mesenchymal stem cells, and here hypoxia ischemia with saline, and here hypoxia ischemia with stem cells. And here you see the staining for IBA1, which is a marker of microglia. So if you induce hypoxia ischemia, you get a lot of inflammation. And if you have hypoxia ischemia treated with mesenchymal stem cells, the number of microglia goes down. And you see this here also differentiated and counted in the two regions. What happens next? We looked at the white matter injury and looked at a functional readout. If you have a convulsion, you have a dysfunction of the brain. Here you see the structure of the brain, and this was measured in FA units. And here you see, if you have hypoxia ischemia, you are losing the complexity of your brain structure. If you give mesenchymal stem cells, this is protected. So the structure is protected. Is the function then improved? Here you see the number of convulsions with hypoxia ischemia, and here you see the number of convulsions if you treat with mesenchymal stem cells. So these stem cells reduce the number of convulsions, so your function is better. You would ask me now immediately, what happens to those cells? Well, we used genetic tracing. These were human cells, so it was pretty easy to detect human cells in a sheep brain, but only 1 out of 10,000 injected cells was retrieved in the whole brain. That was pretty sobering up, that after several days, those cells were completely gone. And then we started to look into the immune system closer. We were struck with the spleen and the thymus that I showed you beforehand. And then we looked at lymphocytes, stained here with CD3. And as soon as you have introduced hypoxia ischemia, those lymphocytes are moving out of the blood vessels into the tissue and initiate an inflammatory response. If you give mesenchymal cells, they stay out of the brain. And then we started to look into the cell phenotypes and look whether these T cells in the systemic circulation behave differently if they got into contact with mesenchymal stem cells. And what we found is that these T cells go into tolerance and are not responding neither to specific stimuli nor to unspecific stimuli. They are more or less hibernating. So mesenchymal stem cells switched off the immune system. That was a completely new finding and completely unexpected. So this is the way how we think about this nowadays. You have a first hit where you start to get an inflammation, and then you have a second hit once your immune cells are being activated. And the good thing is, if you give mesenchymal stem cells, you inhibit on both arms the inflammation. And that is the magic cure that the MSCs can do, and not the differentiation into neurons. So we discussed this morning also cells that they are communicating with each other. And here you have exosomes, or how they are called nowadays, extracellular vesicles, which contain proteins, DNA, RNA, and also lipids, because they have a lipid layer here around. So a highly complex biochemical messenger system that is used in a paracrine way. But it can also be used in a systemic way when exosomes actually enter the blood circulation and then are transported to very remote areas. So we ask then the question, what is the magic that we have seen? Is it the cell itself, or is it the exosome? And if you think about this in a conceptual way, cells have a complex sensing and response system. They can be directed in their cell migration, and they have a decision-making property. They can proliferate, activate themselves, or die, and they have a closed-loop autoregulation. And if you have small molecules or biologics like these extracellular vesicles, they have a molecular recognition, diffusion and transport capacity is controlled, and you have one fixed biological response that you give at the moment of administration. So we went on and did these mesenchymal extracellular vesicles, or exosomes, and here you see the number of seizures, because that is the functional readout. And here you see if you give exosomes alone, the number of convulsions is significantly reduced. So you get this therapeutic effect without having cells in there, just the exosomes are sufficient. That was a big surprise. So the idea that is nowadays considered to be the mechanism is that these mesenchymal stem cells secrete lots of growth factors like vascular endothelial growth factor. You have heard about that this morning. Growth factor like insulin-like growth factor 1 or nerve growth factor or EPO, erythropoietin, that have multiple effects on angiogenesis, on inflammation, on the dying of the cells, also on the stimulation of differentiation and proliferation. So it is these molecules that make the difference and not the cell as such. So now you understand why I am a big fan of animal models. We have now understood what we are doing in these, and you understand why this is one of our favorite things. I should introduce you to Leonie here, for 500 grams, and now at this age. Please have a look carefully at this picture. Do you recognize anything that is not normal? You adopt her. Yes, she's cute, I know. So let's have a look. Look how she holds her hand and look at the position of her face. She's suffering from a mild motor neuron defect, and she has a somewhat adverse neurodevelopmental outcome. She is very much suffering from that because she cannot play the violin. Well, if you are born in this situation and you want to play the violin, I, as a doctor, are very happy with this outcome. She can move, she can talk, she is well integrated in her school, but she feels that she has paid a price of being born prematurely and that she cannot play the violin. So even with this, at the first sight, favorable outcome, we have to look more carefully how we can get back on track of this neurodevelopment that is affected. So in order to keep the time, I wanted to wrap up. This was my content. Now we make this into a summary. Mesenchymal stromal or stem cells are source dependent and the biological function is leading. So I did not discuss about cord blood origin, Wharton jelly origin, or bone marrow origin, but this also plays a role. The basic idea is very promising, but the essential questions remain unasked. Which dose, which application route? The first studies are out, a long-term follow-up is needed. If you know that your baby is doing better at 2 years, you are happy. If you have this child for the rest of your life, you say 2 years is nice, but what about 6 years, 8 years, 18 years, 20 years, 40 years? This will be the future. So is it the magic bullet? We have trusted so many things to be the magic bullet that I suggest some caution, and that we have to work really hard and consistently to find a better helping hand for those preterm babies. Thank you very much, and I want to thank my collaborators. Oops.